would suggest that we start. So let us start then. So hello everyone, welcome to the webinar. So very pleased to have Shawn and Onno today in talking about field phenotyping. So we have a dedicated session on field phenomics. Some background sorry, wrong slide. So some background information on the organizers. So we have the webinar series. It's meant as it's organized by EPPN, Emphasis, IPPN, those networking activities in plant phenotyping, which tend or want to provide an exchange platform for the community to address relevant topics in phenotyping, with specifically also seek contributions from plant phenotyping related work from basically all fields related to this field, and you can submit an abstract. It is still open to submit an abstract, and based on this abstract, so it's a conference-like approach, we can also select this contributions and set up presentations or webinars. We have two different formats, so single talk and thematic sessions. Today we would have thematic sessions having two experts talking about field phenomics. Yeah, and the webinar series takes place every Friday at 2 o'clock Central European time. So it's organized by the IPPN. IPPN is a network connecting plant phenotyping centers, industry all over the globe, with the mission to integrate this regionally, globally fragmented activities and bring all the people together and provide a platform to talk, to discuss, to exchange in a very broad sense. EPPN, another project, it's more specific. It's providing access to the user community. Sorry for the wrong date there. There are calls for access, so you can apply for access. Actually, you could apply. The last call is already finalized, and there will be the last evaluation round is in progress. So in general, it worked very well. We will be able to do about 150 experiments on plant phenotyping. I'm pretty sure, and that was a very successful approach with EPPN 2020, that within Horizon Europe, the new framework program in Europe, that there will be new opportunities to continue with this kind of opportunities. And finally, there is also Emphasis, which is specifically supposed to provide this long-term sustainable development of phenotyping services. So specifically providing access to phenotyping platforms and other services, data, enable harmonization, but also develop training approaches in a long-term perspective, so in a sustainable way. Based on that, we have also a platform. So the webinar is right now, you are sitting live in a stream, and we have also colleagues in China. So there is another platform where the presentations will be streamed, and we have usually quite a lot of participants in there. So thank you for doing that. And as such, to keep it short from my side, I would like to move on now to our presentations from today. The first would be Shawn from the University of Barcelona, talking about proximal RGB imaging for field phenomics. And then Onno from the Research Center in Jülich, focusing more on photosynthesis in a field. And as such, without much talking, I would move on now to Shawn. And basically, Shawn, the stage is yours, and you are welcome to present the proximal RGB imaging for field phenotyping.
Okay, thank you. Just let me get my presentation set up here. There. Okay, I'll share my screen, the slideshow. There we go. How's that? Everybody can see it?
Yes, thank you.
My name is Shawn Kefauver. I'm a professor at the University of Barcelona, like a lecturer professor. Also a drone pilot, and do a lot of field phenotyping. It's been exciting field to be in. I started in field phenotyping about 6 years ago, actually. My PhD was in ecology and remote sensing at the University of California in Davis. And I'd always done some agricultural applications, previously some collaborations with NASA, but these were airborne applications. So the best spatial resolution you could hope for was about a 3-meter pixel, or, you know, we got down to 1-meter pixels back in the day. I hadn't really come to phenotyping until I moved to Barcelona, and I changed to the University of Barcelona. Anyways, this is the picture that I submitted to the photo contest. They told me I was a finalist, but I didn't win. You can see it's a little bit grainy. It's kind of an old camera, I think. Anyways, so this is me in action in Africa here. So I'm going to talk about. Let's see, why is this button not working? There we go. So I'm going to talk about proximal RGB imaging, and that means both in the field, as if you're holding a camera with your hand, or also from drones, which is pretty close and not long distance remote sensing. The research aims and goals of RGB imaging. So we would like to improve data quality and quantity. Phenotyping is often a numbers game, so we can improve both of those. That's good. Using proximal-based, very high-resolution RGB imaging, mobile platforms, meaning basically mobile phones or something that can move, even yourself, and UAV-based high spatial RGB imaging. So what is the purpose here? So we want to improve the data collection consistency and the efficiency of it, which we all know is important for phenotyping. So reduce time spent in routine assessment tasks, reduce the resources spent on non-representative trials. So this was something that I hadn't thought about as a researcher, because we always have these small trials that we manage really well. We started working with some companies, and they had trials all over the place. And if a trial suffers some damage due to a storm or some weather that really ruins it, then they just pull their resources out there. They stop monitoring it. They stop taking care of it, and they improve their cost effectiveness by monitoring trials early on to know if they're going to have something that's going to be meaningful, because they want to go all the way to yield. And then also improve the accuracy of manual quantitative tasks. So real quick, I'm going to go over scales of observation and application, a little bit about the concept of using RGB and color spaces and image processing, and then some examples of applications. So these are all open access, freely available and downloadable links here. They're all works in progress. So if you want to test them out, if something goes wrong, let us know, and we'll try to make them a little bit better. So here I'll be talking about the serial scanner, the fusion and serial fusion, and the mosaic tool. Okay. So scales of application, we've done some work, and we've been able to publish research based on RGB phenotyping at the leaf scale. So flattening single leaves and looking at what we can get out of color quantification there. At the ground scale, you're just holding the camera with your hand and taking up close pictures, as an example of wheat. And also the aerial scale. You can see here on the right, there's example of a rain-fed wheat trial, and the next to the irrigated wheat trial, with a couple of people working in the field there for reference, right? So RGB is in the title of the talk, and what is that? That's red, green, and blue. So if you want to think about these in terms of pretty pictures, but when you want to think about them more than pretty pictures, what we have here are broadband visible light reflectives, which can be meaningful data. If you consider one, they have excellent color calibration. So these are just a plain digital camera. It's got an incident light sensor. It's got good color calibration, because obviously nobody wants to buy a digital camera that takes pictures that don't look like the human eye. They don't look nice. And so they get these color balances pretty well. And we did some testing of color calibrations, and compared to a color panel, one of our publications we put out, usually, R squared is a 0.9 or higher compared to a technical color panel. But also here, you can see on the left, I added in some graphs of photosynthetically active radiation. So if blue is about 400 to 500, green 500 to 600, and red from 600 to 700, you can see that we're capturing information that's relevant here for the quantification of chlorophylls. So if we had a multispectral sensor, we could pinpoint all these different little peaks, and we can measure them separately, but we can quantify the total amount of pigments fairly well, just looking at this combined photosynthesis and absorption. Right, and then lastly, we have very high resolution. So we're talking about centimeters or millimeters, depending on how far away we take the camera and how close we are. Okay, so I have a couple of quick results just from some of our previous studies to give you some examples. So here we go, biotic stress assessments. This was lethal necrosis in maize. You can see the maize lethal necrosis indexes, which are visual scores here, and we're looking at doing indexes of green area. So just a quantification of the pixels at either green or red. So photosynthetic area. We can see the difference of those there. And we compared that to yield, and we compared it to the correlations. You can see here some of these different RGB indexes using alternate color spaces or calculating the green area, such as hue, A, B, U, and V are from the C-Lab color space. Green area and green area are just counts of yellow pixels and green pixels. And we compared that to NDVI, the green seeker NDVI sensor. And you can see that they actually have similar performance compared to some of these other scientific sensors. Right, this is an example of yellow wheat in rust. So again, comparing some of these hue, A, U, green area, and green area RGB indexes to the performance of NDVI as a field sensor. And you can see the correlation with yield can be equally high or even higher using just a plain digital camera compared to this example of a scientific sensor, NDVI again. So we know that NDVI isn't the solution to everything, but it's a good point of reference. And I know a lot of people are excited about getting NDVI in their field trials or some scientific sensors. But actually with the plain digital camera, you can do just as well or better depending on the growth stage, right? Here's another example of nitrogen stress in maize. Again, we were looking at correlations between here, these are the leaf chlorophyll content with grain yield. And then if I can get a little pointer somewhere, spotlight. How does that work? Oh, sorry. Here, this is an RGB index, green area and green area. Here and pretty good correlations with grain yield in total. Oh no, I have to get rid of that. I'm going to change it. There we go. Oh no, the picture's still there. The drawings. So anyways, and this is another example of water stress in wheat. So again, at the aerial scale, this is a little example about the changes in resolution. So comparing RGB indexes to NDVI, when you're flying on a drone, if you have much lesser spatial resolution, then you might see that that from a drone, the NDVI might function slightly better. But if you compare it to the ground scale here, when you have higher resolution, and I think basically some of this is because with very high resolution, these indices based on just photosynthetically active radiation coverage and in the broad bands have a very high spatial resolution, and that helps them keep from being saturated during dense canopy cover. Here's one more about nitrogen treatments at the study we did in Spain. So I get my eraser here and I'll erase those lines. There we go. So you can hear comparing greenness to grain yield. Sorry, my screen just went black for a second. Comparing greenness to grain yield had a good correlation, especially here looking at barley plots and looking at nitrogen content. Also early senescence, here the crop senescence index related to grain yield. And these correlations were as good or better than what we got from our multispectral camera, looking at the more advanced soil adjusted vegetation index or the optimized soil adjusted vegetation index. There. So those are a couple of quick examples showing that yes, they do function. So moving on to something that's a little bit more practical, we thought, well, if this would be easier for other people to use, maybe it could benefit the community. So this was something that we thought about giving back. We teamed up with a company. We started working on RGB field-based phenotyping for wheat for cereals. And people ask me, will this work for other types of crops? Is this the cereal scanner? And I said, well, you know, there's actually, out of all of this, there's only one index, which is actually specific to cereal crops. The rest of it are just traits that are be interesting. So moving along, and we published also, this software is available for free. We also published a Journal of Visualized Experiments video publication that's also quite interesting here if anybody's interested in that. That explains how to use these different functions, how to collect data in the field, and what sort of equipment we used in this study. So this one is early ground coverage or early vigor. So it's just a higher accuracy segmentation and reliable segmentation of percentage of ground cover that's sort of been optimized to detect vegetation or not vegetation for very small leaves and shadowed or sunlit leaves. So we're using here the Sea Love image color space. So we started exploring how professional photographers were calibrating their color, and they often transform them into different color spaces. So we use the Sea Love color space, and it's interesting because it separates color from brightness or luminance. So we can segment out what's green regardless of whether it's dark green or it's really bright green. And so that improves the accuracy over just using red, green, blue, green reflectance to separate out soil from something else. That gave us good results. We used it over the entire field season, not at just the beginning of the field season, to look at some low cost potential for assessing grain yield using RGB images. And then we developed, which was a little bit more involved, an ear counting system. So this is something else for trait-based phenotyping that was something that the companies and other phenotyping breeders were interested in. There was some debate about whether counting the ears in a crop like wheat or barley would actually have a good correlation with yield, because there's always some compensation mechanism. Sometimes you have smaller ears, but then there's more of them, or you have bigger ears. But it's definitely something that most cereal breeders are doing in the field, and it takes a long time to do accurate counts in the field of these, because we're looking at hundreds of different ears. So we, and this is from our Jove publication, also some more details or approach how to implant methods, where you just take an image. We have to record in order to get ear density, or number of ears per square meter. We need some information about the camera, how far away you are, and also the zoom, the lens you have, and the resolution. But we can run it through this ear counting pipeline, where we filter out unwanted noise using location and median filters, and then we can count the number of ears there. Similar techniques have been applied using convolutional neural networks that are trained and functioned as well as, or sometimes even better than our technique. Our technique is sort of based on knowledge of filtering out unwanted aspects of an image step by step, and then getting to what you want instead of using sort of a black box algorithm. We had fairly good results. Of course, the results fall off a little bit if you try to use this function on a crop that's not counting ears, like corn or something like that. It's not designed for that, and we don't really know what sort of results it'll give you. We also sort of try to specify a certain range of crop growth and senescence. Obviously, there's one moment in the crop growth phase where the ears are still green and some of the leaves start to yellow, and you see a big color separation between this. We also scaled this up and tested it. So if you have a drone and you can take pictures at high enough resolution or fly low enough, then you can also do that. One word of caution from experience in the field: if you have a very large drone and you fly close to your crops, your drone will turn into a giant fan and your crops will move around too much and you won't get any good pictures. So we tested out flying a really big drone 10 meters away from our crop, and all the pictures were terribly blurry. We later figured out why. And so this is sort of a big schematic. This is the explanation of our maturity estimation, where, and this was launched in our maze scanner, which was part of a collaboration with CIMMYT some years ago, which was using this selection of algorithms called BreedPix, which was developed by Jaume Casadesús when he was at the University of Barcelona a couple of years before I joined the teams here. And he was the first one who started using the different color spaces. And he was just taking out the average values of these different color spaces, the hue, saturation, intensity color space, which is where he made his green area and green area indexes, which capture the range of yellow and green pixels, or the range of only green pixels. Taking that, Mainasara Zamanala from CIMMYT, in a collaboration with the University of Barcelona, developed the crop senescence index, which is the ratio actually of green only pixels to green plus yellow pixels. So that's the crop senescence index. He's looking at the different color spaces, and then I also saw some interesting indexes out of the USDA. So the triangular greenness index, which calculates the area of the triangle forming the deep valleys of red and blue with the peak of green, and the normalized green difference index, which is similar to NDVI but moved over to look at the difference between green and red. There have been a lot of different RGB indexes launched recently, and I recommend that you explore it a little bit. We're going to try to actually start adding in all the different indexes we can find in the literature to these tools, so they're a little bit more user-friendly and that they support more than and what's actually out there in the scientific research. So this is the example of the crop senescence index, and it actually works quite well. You may have an inverse correlation with grain yield because the value goes up as you become more senescent. And so if you think about early assessments of chlorophyll content or early assessments of vigor, people are going out and visually estimating senescence. And here we can do that with a quick picture, and it's more quantifiable and it's more reliable. So we took these ideas from the maze scanner and the serial scanner, and we thought about making other tools would be useful to support people in collaborations. So we started playing around with the Open Data Kit Collect platform, which functions on an Android phone, and it allows you to create a questionnaire on your phone, and then that connects to a server which you can develop. And then we developed a couple of tools that would process the data that you upload to the server and then append that shared database with results. So there's some more explanation about it on our website. But Open Data Kit's a fun tool. And we used it testing some Earth Engine things in a project here at the city of Barcelona, monitoring LAI changes in the parks, and also with the FAO at one point in time. So these are just PC-based GUIs where you can put up the location of your Google spreadsheet. It finds that Google spreadsheet, it downloads the images, it processes them depending on what sort of analysis you select, and then it appends the result to that shared Google spreadsheet. We thought this was interesting, especially for wheat ear counting, because the algorithm is a little bit computer intensive. If we put it on a mobile phone, we probably end up killing the battery of the phone. But here we just upload the data. It's what I sort of call cloud processing light. So you can run this on any computer with a PC connection. They're Java-based. So any PC with an internet connection can also be set to continuously process the record. So it'll just download, process that data, and append the results in real time. Also wanted to talk a little bit about using RGB cameras on drones and how it creates some new challenges, but also some new benefits. So one of the good things is that a lot of pre-processing or post-processing that's required with these can be completely semi-automated. So you fly your drone, you take a lot of pictures according to a flight plan with overlapping images, and then you can create a mosaic out of it almost completely automated, which is quite nice. And a lot of different softwares out there and cloud-based support as well. And then image extraction. So having done large-scale image analysis first, I was surprised that in phenotyping, we want to actually process every single microplot separately. So the first year, we flew our drone a dozen times, and we spent 9 months manually cutting out all the little microplots and images to process them. Right. The pre-processing part was fairly quick. You put it in thanks to the structure from motion mosaics. You put it in, you run it, it produces not only a mosaic, but also a dense point cloud that you can test, you can classify. Now this does take a pretty good computer to run, but it's largely hands-off, and you can get some interesting data sets. You can fly however you want. I was just manually flying this drone around, and it produced a perfect orthomosaic for me. And this is some examples of a little zoom in. So you can produce a dense point cloud. So looking at these crops in 3 dimensions, you can classify it as ground points or crop points in order to remove background pixels based on plant height, which can be interesting if you have a lot of weeds in the field and you want to measure only the crop, which is so high. Here, this was a maize crop. It's a lot of fun to play with too. I don't know if I mentioned that. And actually, if I'll just throw this slide in because it's a really funny picture of me. Actually, I don't know where I got that hat from, but we had a bunch of projects where we said that we would fly drones to collect data, and it would be great because we could phenotype thousands and thousands of plots. And then right before the project was awarded, the drone laws changed, and it was made illegal to fly drones in some of these countries. So we ended up with a camera on a very high pole running through fields, taking pictures, trying to do drone simulations. You can still get good results from it, but it's not quite as sexy of a science. Anyway, so we're doing modified cameras and RGB cameras as well. So we developed the mosaic tool. It's not really high class, but it just helps you automatically segment a lot of different plots. And so this allowed us to get up to segmenting out, cutting out about 1,000 plots an hour from when we had lots of phenotyping work to do. And so you just specify the area of your study, you specify the number of plots in a row, the size of the plots, the number of replicas you have along beside each other, and it'll draw you a nice grid of whatever, 500 little squares. We have, then there's a step in the middle where you can manually adjust them if you have some errors in your plots. And then you export them out as what we call micro rasters. So every micro plot has its own little image, and then we can batch process those, and we included a bunch of different code for batch processing. This is also something that's a work in progress. So we're going to try to add in more indexes and support for other different types of sensors coming soon. Here's a couple of examples. So coming out of field pictures here, you can see the results of an image of hue of the green area, and then also taken from the ground at higher resolution, and then from the drone as well. So a little bit of loss of resolution, but actually the results were quite similar. This is an example where we cut out several hundred different plots in a row. We were comparing some of these things to field sensors such as SPAD chlorophyll or the NDVI with the green seeker. And you can see there's definitely a loss of resolution from a drone, but actually we get still quite good data. Here we go. So this was my, this used to be my conclusion slide, and now it's just random thoughts. So red, green, and blue. For phenotyping, there's always that toss-up sort of the difference between resolution, precision, or throughput. So if you don't have that much funds to play with, you can play back and forth with these. So get higher spectral resolution data, such as visual, near infrared, multispectral, even thermal. be substituted for very high spatial resolution RGB data. So can you substitute spectral for spatial? To some extent, I mean, looking at infrared in NDVI, sure, that you may be able to get similar information, but these are broadband indexes. I think at some point in time, if you want to do narrow band and look at specific pigments, you're not going to be able to get to that point with RGB. We have found that it has limits. We have been testing with a 45 plus megapixel camera. Obviously, sometimes spatial resolution when you're flying on a drone can be getting near the threshold where you start to lose quality of the results, but it works out pretty well. Also, can you exchange temporal resolution for spectral resolution or for spatial resolution? So if you collect in a phenotyping program, if you have something that's so quick and easy to process, can you do that? Are UAVs always the best option, or can you just run through the field and take pictures of every single little plot? That's certainly quicker to process, easier to collect, though maybe not as exciting for some people who really want to fly a drone. And then, different optional band collection. So as far as future ideas and where to go with this, I think there's a lot of information in RGB cameras at the high resolution. And so the idea is to talk with breeders about what they want and how we can help them get there with better data and faster results. So as the Beatles once said, I always get by with a little help from my friends. So of course, I'm just a professor in a large group of people with research projects all over the place. This is the Integrative Crop Ecophysiology Research Group at the University of Barcelona, led by Jose Luis Arauz, and also all of our collaborators around the world and in Spain and everywhere. So that's all. Thank you very much.
Thanks a lot, Shawn. That was a really, so what was that, Jose Luis, the last picture in thermography?
Oh no, that's me. That's me. I'm flying the drone and walking in front of it, filming myself at the same time.
Oh, okay. Wow.
So it's sort of like walking and chewing gum, but for researchers.
Yeah. Yeah. Thanks a lot. It was a really nice presentation. Nice overview of your work. So there is a question in a chat. How do you manage light conditions in general? For example, in dense canopies for sunny and shady leaves spikes.
Yeah, so that's a good question. So actually using these alternate color spaces, since they separate out color from lightness, that allows us to quantify color, whether it's green or not green, or separate from the sensitivity to lightness. I would like to add also that all of these commercial RGB cameras have incident light sensors, and they have built-in processors, and they adjust the contrast and the lightness in real time. We do recommend taking pictures scientifically, which, for remote sensing, we always try to collect our data within a couple hours of solar noon. So actually shadows can still be a problem. And we recommend, if it's sunny, to take your pictures around solar noon so you have fewer shadow effects. The camera will adjust to the brightness, but the shadows can still be capable of masking things. And actually, some of our best results have been taking pictures all day long on a sort of uniformly cloudy day. So since it's broadband reflectance, that small changes, the quick response of the incident light sensor means that if you're taking images in diffused light, you have no shadows, and the results can actually be better.
Mm-hmm. There is also a question, what is the resolution for your UAV? Take disease evaluation, for example. How high does it fly, or does it have to fly?
How high does it fly? Well, yeah, so we like to fly our UAVs because otherwise they're quite heavy if you walk around with them. We have several different UAVs. We've been testing. We've been very happy actually with the DJI Mavic 2 Pro, which has a built-in 20 megapixel camera. Earlier on, we were flying just a regular Panasonic 16 megapixel camera, and that was getting pretty good data. We even tested a 43 megapixel Sony camera with an integrated GPS on it at one point in time. And that really collected a lot of data, and it took us longer to process it just because the images were so big. But the point of that, I think if we could fly at 50 meters, we could cover a lot of area, and that gives us, I think the minimum resolution on the ground spatial distance for us was about 1/4 of a centimeter in order to be able to count wheat ears. So we did a simulation study lowering the spatial resolution. So that means if you had a 20 megapixel camera, you could fly like a Phantom 4 or a Mavic 2. You could fly at 25 meters and you could still count the wheat ears with pretty good accuracy.
Mm-hmm. I would have a question on your, so you already mentioned this light issues when there are different sun angle and those kind of things. We are right now discussing about the data comparability, reusability, all this fair data. So basically reusing data from all over Europe, all over the world. Is it possible to use data from different facilities? What would be your recommendations when you want to link data from different field experiments, assuming it's on even on the same crop, potentially the same traits you want to extract? What would be your recommendation to enable this reusability of data? What would you need to take into account? Yeah.
With an RGB camera, one thing that's nice about them is a lot of them record quite a bit of extra information in the EXIF file attached to every JPEG. You know, it would be nice if people would collect their images or store them in a lossless format. You know, JPEGs can lose quality over time. They're stored and they're compressed and decompressed. But also the EXIF file attached to most JPEGs that come out of plain digital cameras will give you the day of time, the hour of the day, the exposure, the sensor details, the resolution, and that's all actually really useful data. It doesn't always include, I mean, there's some cameras that are starting to provide more data, but we did one study where every collaborator was sending us pictures taken with a different camera, and actually the results were pretty comparable. Some of the cameras we were surprised that they started using such a low quality camera when we suggested that they got something with a large sensor. You really see the differences if it's a small sensor camera versus a large sensor camera. You know, I'm always surprised when somebody has a mobile phone with 25 megapixel images coming out of it, because the sensors is itty bitty, teeny tiny, and they're doing some sort of black magic there to turn it into a 25 megapixel camera. But actually, if you have a large sensor that takes, like a micro four thirds with a 16 megapixel, that's a fairly large sensor for that number of megapixels. And so you have, I think, better quality data in there. So that would be my sort of recommendation, is try to get a large sensor camera, and then they're fairly comparable. Oh no?
How far do you think, if we're talking about data, that we can get your data from the drone in direct communication into an action of the field? So that means there should be some processing hub in between. That, or do you have already examples of when this is executed?
You mean real-time transmission?
Yeah.
Well, everybody talks about that when 5G comes around. We'll be sending it off immediately. I think cloud-based real-time processing could make sense. It depends on what sort of processing you want to do. One of the things I think is interesting is these deep learning algorithms are sort of famous to take a long time and a lot of computationally intensive to create, but then applying them is actually fairly quick, because they really are good at narrowing it down to getting good results fairly quickly. And so something like that, I mean, you could be flying a drone over, or you could have cameras on a tractor, for example. They could be counting wheat ears and adjusting the application of fertilizer as it goes, based on the detection of the density of ears as it goes over something. I don't think that's too far off. I mean, there's a lot of data there. I think we're still in the phase of collecting too much data and not knowing exactly how to process it and give decision support in terms, if you think about going on to precision agriculture from phenotyping or something like that.
From a phenotyping field that I would be, for example, interested in plant height, which you showed to derived. Would you think that's feasible?
Well, I wouldn't say with RGB, but now there's a lot of lasers, laser sensors that might have better precision. RGB, so it's quite computationally intensive to create these three-dimensional models, even on big processing machines. But plant height in real time, I mean, I think the technology readiness level is there. I don't know about doing it in real time because it takes a while to process and create these dense clouds.
Yeah, thank you.
Thanks a lot, Shawn. I suggest then that we move on to the next speaker. So it would be Onno now. Maybe a word also to all the participants. So please feel free to put your questions throughout the presentations into the chat. We will have also afterwards, after Onno's presentation, some time to specifically address the questions to Onno's talk. And afterwards, you can pose also questions to both speakers. In general, whatever you think on getting feedback from field experts would be required for your research, what you are interested in. So please feel free to put everything you think you would like to know about field phenotyping into the chat. So as such, I would then suggest that we move on to Onno. And so Onno, research from the Research Center Jülich. But Onno, please introduce yourself shortly, and the stage is yours.
Thank you very much, Roland. Indeed, my name is Onno Muller from the Research Center Jülich, where I'm since 6-7 years working in field phenotyping. And I have a background in plant ecology, and as such, and also measuring photosynthesis in forest, in warming experiments, and also in very small scale level, connecting that to phloem transport. But today I would like to talk with you about proximal sensing of photosynthesis by plant canopy fluorescence for field phenotyping. And Roland may have said it in the first slide, or it was announced. Today, this is a field phenotyping session, and it's also the last 2020 IPPN webinar. For field phenotyping, actually, it's timely because, well, we haven't had snow yet, but most of us, in terms of in Europe and the western half of the world and the northern half hemisphere, our plants are harvested, and we have a slow downtime to start analyzing data and actually look at these webinars and presentations. For that, I looked up webinar again in terms of what's the webinar different from where we all usually meet on our conferences. And if I look at just one dictionary explanation, is the educational presentation, where you can submit questions and comments. So I'm very much looking forward to your questions and comments. And I will do my best for education. I think every presentation in itself is educational, but yeah, what I want to do is bring you to the field, also with some pictures and also with an outlook. That can be something typical to the end of the year, looking into the future. And also here, we had the excellent talk from Shawn previously, where he positioned drones over the field in different locations. If I think a bit further in the future, I will be looking in interaction of these drones and multiple phenotyping tools on the ground that can measure what a drone cannot, because we have limited in weight and not all sensors can go there. And ground vehicles could potentially also put more actions beyond phenotyping. As such, it may look like a farmer, but it's the normal people in the field want to get the information, but also want to contribute to the field. But as I said, this is a bit outlook in the future, and this is not. I would like to narrow down. Otherwise, we pass these 20 minutes by far. But I would like to point out that it's actually, this vision is not too far. And if you want to know more about this vision and where these slides come from, look at the bioeconomic review of in Germany, where they try to put from this area where the Forschung Center is located, to put a model region for the area to really implement all these technologies into the field. And this concept presented here actually may be reality in less than 1 year time. What I would like to talk with you about is specifically field phenotyping in this talk. I would shortly introduce a pure chlorophyll fluorescence methods from leaf, field, and proximal level. Say a few more words in detail about the light-induced fluorescent transient. That will be the heart of my talk. And then use examples of the LIFT, the light-induced fluorescent transient method, in proximal sensing of heat and drought in durum wheat, elevated CO2 in winter wheat, and a cercospora infection in sugar beet, where I will show pictures of the field and also try to bring you to the field. Although we meet here online, just look very closely, and I hope we can see and come there together. First, a familiar slide for some who looked in field phenotyping before. I've shown at MIMO quite a lot of times the 5 pillars of field phenotyping. Biologists and all people interested in nature, I think we have in common, we want to understand traits, be it from plants, animals, or other living creatures. And understanding these traits, we need a sensor that can quantify it. These sensors are developed rapidly, often when new techniques come available. Think of these cameras, think of the drones that are recently there, and the computing power coming with it. But you also have to put them at the right place. Shawn elegantly showed that when his drone was not allowed to fly, that they created went for the phenopole, because they said, yeah, I don't have my position, I don't have my drone anymore, it's over. It's thinking about where to put your sensor in the right place. And yes, that may require a pole to walk through. To answer questions, experiments is essential where we all meet together to quantify our traits. But it's not just walk in the forest and say like, oh, what is photosynthesis doing today? Often you especially in the phenotyping world, you have a more controlled breeding experiment. And last but not least, we quantify the environment. Phenotyping, as you all know, is environment times genotype. And often we focus a lot on sensing positioning system experiments of getting different genotypes and the different quantification of these phenotypes, but in relation to environment. So we really need to measure at high resolution also environment and get these data out. Today, not much on how to measure environment, but measuring about chlorophyll fluorescence. Chlorophyll fluorescence, a trait measured for quite some years already, and the leaf level measurements have been really exemplary for that. You may know the Waltz me pumps, or even the Photosync handheld affordable devices, where if you look at the website, you do not find anymore actually how chlorophyll fluorescence is measured, but it's all about what you can do with a trait. But both have been really increasing the wide use of fluorescence to quantify photosynthesis processes. I think that's the key message. But for today's webinar, they are actually too close leaf level. When I talk about proximal sensing, I want to go fast. I don't want to clip something on a leaf and go to the next plant. So going to the other end, you saw a similar analogy in what Shawn introduced, the field level. We have for chlorophyll fluorescence in our group, especially with Uwe Rasche, developed hyperspectral sensors that can, at a field scale, up to 1 meter, 3 meter pixels, quantify sun-induced chlorophyll fluorescence. That led to a 300 meter scale, a flex satellite that will be launched in 2023. We did not include that in today's talk, also the level where at a 1 meter resolution, can we use this sun-induced fluorescence for phenotyping? I mean, where we compare different plots with different genotypes, and also handheld sensors for sun-induced fluorescence. So it's not out of the league, but sun-induced fluorescence, it's in a way very applicable, non-invasive, but at the field level, it's a little bit too remote for this webinar. So what are we going to talk about? Well, I would like to now talk about, as I said, about chlorophyll fluorescence, then laser induced. I'm very happy that Roland is actually our moderator today, because he was one of the guys who carried around a laser induced fluorescent transient method, a heavy laser system up to roofs and into towers to tried to quantify, and he actually did in the field, chlorophyll fluorescence. He compared that. That's the graph here, the figure on the left side. He actually empirically showed, like, if you measure with your lift measurement, the light-induced fluorescence transient, or with PAM fluorimetry, both correlate linearly with your gas exchange. So we have a comparable method. Either we do it with light-induced fluorescence transients, or we do it with pulse amplitude modulation, or we do it with gas exchange. We get to a comparable photosynthesis measurements. There are also other developments in this direction, other light-induced fluorescence or laser-induced, I must say, fluorescence measures that please have a look at it. But they're not all got commercial, and one of the limitations may be that they're often heavy, and actually they are high danger for your eyes. You see Roland is wearing glasses nowadays. But it's one of the solutions. And also the only commercial solution I'm available is a light-induced chlorophyll fluorescence units by the company Soliant, where they put, instead of a laser, an LED. And you see here on the left side an image of this white box. The earlier ones were black, and when they turned white, we start to understand also better what's going on inside. I mean, again, we being it now an LED instead of a laser, we compared, for example, the lift derived photosynthetic efficiencies with PAM fluorimetry, and we show a linear correlation recently in another context, but looking at non-photochemical quenching in different Arabidopsis varieties. You see here on the left side displayed with open symbols PAM non-photochemical quenching, and with closed symbols lift non-photochemical quenching. And you see they follow exactly the same pattern, and the difference between the symbols are different mutants inhibited in their non-photochemical quenching, indicating that we have a method in hand that can quantify fluorescence for us. So how does it really work, the lift? You may not all be familiar with it. So the lift protocol is doing nothing more or less than putting out a series of high frequency blue light flashes that separate the first quinone acceptor so that it's fully reduced, and you get a maximum in your fluorescence signal. And after that, they send out a reduced in a reducing rate flashes, and you have a relaxation of your fluorescence signal that is related to the electron transport chain processes downstream from quinone A to B. This last part of this so-called altogether, that's called the transient. These are traits that were not possible to measure so far, whereas this early stage is getting to a maximum fluorescence as an analogy to PAM fluorescence. So in another display of actually real data is showing here again the terminology and lift traits that we nowadays use for phenotyping, which are the photosynthetic efficiency, FV over FM, or as it's slightly different, the method, we named it FQ from the quinone acceptor over FM. And we have from the maximum chlorophyll fluorescence, and we have the slopes of the relaxation. That I will not go in exactly the downstream processes of the electron transport, but I would like you to have as a take-home message that we here have a method that resembles the photosynthetic efficiency measurements as other tools that we can now measure in the field, but we also have additional measures in the downstream that can measure the electron transport rates downstream which are thought to show the first step from the QA to the peak plastoquinine pool in 1 milliseconds, and the next step from the plastoquinine pool to photosystem 1 that happens in 2 to 20 milliseconds. It's a full transient. I talk now about the relaxation, about the maximum, and about the steady state fluorescence. But the full transient contains more information that I will leave out for this talk. And I will leave that out also to a distinguished audience shown here on this slide, and also with the publication lead. I must mention specifically Barry Osmond, who had a huge interest in this remote proximal sensing of fluorescence using this terrestrial lift, and he is actively working. He just submitted another manuscript on the initial phase, for example, of the fluorescence transient. But I would like to take you now into some examples of the field, and specifically some pictures to illustrate, such that we have an impression, how did we do that? How did we do the phenotyping? And also maybe guide you or help you to see some pragmatic solutions that we may have taken. First example, as I said, was heat and drought tolerance in a derived panel. I start out a bit tricky, but I start out in the greenhouse. Also to remember you that often these large panels are phenotyped in greenhouses, not so often in the field because methods may be limiting, but also in greenhouses, it's scanning calls for pragmatic solutions. Here you see the lift, a black version of it, and here you see the large collection of derived panels we investigated, and of the source of Roberto Rosa of the University of Bologna. We were there in summer, and in summer, in addition into this greenhouse of being warm in Bologna already, we turned on the heater. And when you turn on the heater in a greenhouse, you have 40 degrees temperature, and morning luckily was still relatively cool for Christoph and Lars, who were inside this greenhouse measuring. But what we were looking for is the photosynthetic efficiency at the hottest moments of the day, decreasing significantly and recovering a little bit in the population of derived. This pattern is kind of expected, but what is the interesting part we see here is a large variation that is in there, because we are looking, well, can we identify in this derived panel some genotypes that show specific response? I picked out some of the extremes. Again, on the y-axis showing the photosynthetic efficiency, on the x-axis development of heat over time during the day, because we were in a artificial heated, but an addition of sun, a summer day greenhouse, and you see in some genotypes photosynthetic efficiency dropped tremendously and still recovered to a certain extent. In other genotypes, photosynthetic efficiency didn't even drop to half of this other genotypes, but the recovery was also less relatively then. So we could show for this derived panel some difference in heat tolerance, mind you, in a greenhouse. We took it to the next step where we, in natural conditions, also actually winter time, no rainfall, we didn't focus so much on the heat, but the same panel we focused on drought, because in the Arizona desert in winter there's not much rain, and with irrigation we planted and established a plant canopy, and then we stopped irrigating. And when we stopped irrigating at the beginning, Nicolas Seminardi came and he went with this lift into these 252 genotypes and measured those. Here you see Nicolas from top view, and you see him also pushing a cart by our colleagues from Arizona through the field. What I kind of like on this picture is on the background, you see the automatic gantry chart with also fluorescence sensors in here that have to close the canopy and really have a covered area that everything is dark. That's one of the advantages. You're measuring a light-induced fluorescent transient that can measure during sunlight with a little bit of a push. If you push, we found the same again as with the heat stress. Also, drought stress reduces your photosynthetic efficiency. This was done in March and the winter, and at day one, irrigation was stopped and the soil dried out naturally in about 2 weeks' time. And during that time, you have a reduction of your photosynthetic efficiency. We were not at all at ripening. Booting was more or less at the end of this stage. So we had a green canopy really decreasing. More important, again, here was the different genotypes. I only choose to show here spatial distribution, but what you can see already, I think, is that you have each little pixel color identifies a little genotype at a period of time. And this is where we then really going into high throughput phenotyping of a large set. and we can locate this to the potential genes or the QTLs associated with this trait, with this fluorescence traits. 1 year we had a drought. 1 year actually was not always enough, so Nicolas asked me to go back a second year. He did it. I mean, I must say, the bike got another color, as you see it. Nicolas got brown again of pushing up and down this bike through the fields, but he collected a lot of data. I may ask you to look this data in detail into publication. It's about ready to submit, and happy to share it in a few months' time with you to talk about detail in these results. For that, I would like to go to the next example with you. Another field, not so warm and dry, but just nearby for us in Germany. That's a winter wheat field. You see it here. And in this winter wheat field, you see a typical what they use for phase structures, a ring structure. And in this ring structure, CO2 is blown upwind, and wind carries CO2 over this field. As such, plants or canopies growing in this ring, they are growing at elevated CO2, about 600 ppm in this area of the ring. Such we can test different varieties, different genotypes, how they will perform under future CO2 conditions. Again, here was our interest, of course, what is the photosynthesis response? How do plants photosynthetic efficiency respond to this elevated CO2 growth conditions? We started off there again with a bike. These rings inhibited a bit driving through the rings, so we made an extension. We drove by alongside with a bike, and we had an extension measuring through the rings with a lift sensor here in the middle. Yeah, you see it is slightly muddy. I kept out a picture of the mud pushing through, but it was quite so a push to get these data out. But the guy, Fikas and Andre Krause, I must acknowledge specifically, they did get at a regular interval at the beginning of the year photosynthetic, in this case, the reoxidation efficiency measures over time. You see that it increased. This is about January to March, April. Photosynthetic reoxidation efficiency increase over time. And if you look if you can see these different colors, white is the control plots, and grayish are elevated CO2 conditions. And on the elevated CO2, this reoxidation efficiency tended to decrease in the most of the season, actually. This I'll come back to that later, because we did again in 2019. We now had the availability of a large gantry system, universal to any sensor to put on. But as you may have imagined, and this talk is about the lift, we did put on the lift as one of the first sensors to quantify all these plots in one ring without having to push the bike alongside. For that, I would like to show you also some data. In this case, a diurnal pattern of the early parameter that I showed already before, the photosynthetic efficiency. In this figure, also light intensities over a day are shown, as well as temperatures. And what you see is that the photosynthetic efficiency decreases when light gets high, and at the end of the day, when light decreases, photosynthetic efficiency increases again. What you also see is similar to the reoxidation efficiency. If you look in very detail, there are some significance levels indicated here. But again, the control at higher values than the elevated CO2 plots in photosynthetic efficiency. If we look in more detail of 3 genotypes out of this population that we measured, you see again the same pattern. We have in red the control, in blue the elevated CO2 concentrations over the day for 3 genotypes. They were in slightly different developmental stages, and NDVI here shown as a reference, where these were greener and these were actually in a later stage with lower NDVIs. What you see also here, at the end of the day, the elevated CO2 plots are higher than the control plots. And for this this variety, over the whole day, the elevated CO2 was higher than in the control. Now you may wonder why, and this is actually where also the nice thing I think about in our starting questions are, how does photosynthesis respond to elevated CO2, and what are the mechanisms behind it? And what we see here, and what we saw in all the previous videos, is that there's a reduction of photosynthesis, and you see it specifically here developing after midday. So when plants have used up all their carbon or saturated, there we know of these feedback inhibition mechanisms, and that may exactly be what is here in place between these two varieties. So photosynthesis, they have a lot of CO2, they cannot export more to their sinks, and the source get an excess of sugars, and photosynthesis is downregulated. What you see in a downregulation also of your photosynthetic efficiency. So that's a lot to talk about, and also interesting material for discussing what are the mechanisms in place in photosynthesis regulation, which also I would happy to do with you again, but as a teaser for future discussions. Last example and another teaser go into the direction of proximal sensor of biotic stress. In this case, the brown spot disease, the Cercospora in sugar beets. This Cercospora really leaves brown spots on the leaf. Can be detected by multiple methods because you could detect the brown spot. And we used again a similar approach. In 2018, we went with a bike and we drove over, and we could see that in Cercospora resistance, different varieties with a high resistance and plants with a low resistance, when they had a low resistance, they had more Cercospora, more brown spots, and they also had a lower photosynthetic efficiency. In 2019, we went in with a robotic vehicle and we saw the same pattern. In 2020, we took a bit more time. We went early on in the season, and later on, we again used our robotic vehicle. You see it here in a fast forward mode, moving over the field. This was not the actual speed, mind you. Nevertheless, although we put in a lot of manpower and a lot of effort, you see that in the third year, this pattern was actually not so clear. So Cercospora was clearly present at the end, but in quantifying photosynthesis, we did not see such a clear difference. We do not have an answer there yet, but it points out also, don't say hooray at point one. Careful, use a few more years sometimes to really quantify your traits. That's my last example that I wanted to share with you. And as such, I call my random thoughts take home messages, but they may be in relation. So what I wanted to show you is that the lift is one sensor you can use for proximal sensing of photosynthesis in field phenotyping. I hope I've shown this with different examples. But also each sensor you may use, but do consider that the plants have a leaf canopy and hardly ever grow alone with one leaf. That is in relation, yeah, I mentioned in the beginning that the clamp-on methods can handle one leaf that is time consuming, but the more I thought about it, it's also, if you clamp one leaf, what do you really know about your canopy that produces, in the end, your yield of your plants? What I said in the slide on the field phenotyping is also quantifying the environment. That's never constant, and it's between seasons, between hours, between seconds when clouds may pass by, but also between years. Do take that variation into account. The plant grows often over many months, and has the year or the environment has a strong influence in that. Position your sensor carefully at all costs. At all costs, between brackets, you have seen some automatic vehicles where you say, yeah, but we don't have that and they're so expensive. But you have seen also that I start typically with a bike, or I go, or I finish with, as long as I can position my sensor on the right spot. You saw a nice example of Shawn, also of the phenopole. So all costs may also be a lot of time and effort of labor to put your sensor on the right place. Do not hesitate to consult us if you anyhow would like to use these big canopy systems or sensors, highly calibrated. Roland already indicated some opportunities that may come up, and successful opportunities that we used in the past. I mean, if we work together, I think we will know more. In that sense, I must also really acknowledge the large shoot dynamics group behind all this work, a colorful group. Very happy to be able to work with all these people. Without all these people, it would also not have been possible. There are a lot of other people working in the research center. Urash and Uri Shure, I must mention specifically of being heads of different part of the institute. Of Campus Canaldorf for excellent field support, of groups outside of our Forschungs or research center in Italy, in Arizona, in other universities in Germany, statistical and field work, and beyond that, and as well breeders and projects such as the data plant colleagues there. With that, I'd like to end my talk and thank you very much for your attention. Time to harvest. I mean, we'll say in the light of a webinar, open to answer or discuss any questions or comments with you. Thank you very much for your attention.
Thanks a lot, Onno. That was a very nice overview about photosynthesis, fluorescence in a field. So there are some few questions. So one question is actually from Philip. How did you account for variations in green biomass for the area integrated fluorescence measurements? It's specifically to the experiment in Arizona.
The variation in green biomass?
Yeah.
Well, we did include actually what I did not mention is that we in the lift sensor we also have a hyperspectral sensor integrated. Philip may have seen on when we used the white bike, I had a picture in there that the white bike and the lift sensor was measuring over a white reference, and this was to calibrate the incoming light for the spectral sensor in there. And as such, we could quantify in the same time as we measured with our lift the hyperspectral signature, and from that we can get different greenness indices of the vegetation. And in our statistical models, we included this variation in there.
How did this look in practice, Onno? Since the lift does integrate over a small but nevertheless an area,
yeah.
Did you have problems, for example, when soil was in that area, or anything other than a plant, maybe?
Yeah, good question, and actually thank you, Philip, for pointing that out, because I didn't elucidate on that the lift is actually an active sensor, and as such it sends a blue light and retrieves back the reflected signal of fluorescing light, and it's the calculations that we do are all ratios. So that is from starting fluorescence in a steady state to an excited state, which all happen on only green parts. In that ratio calculations, the soil covers are not taken into account, contrary to the spectral sensor that is built in, where indeed you have an average or a mean of soil and canopy cover, which we did not exclude for. But the lift sensor specifically only looks at the active chlorophyll tissue by the method itself.
Oh, okay, thank you.
Yep. Thanks a lot. Thanks for this. So there is a question also about how does the environmental factors affect your measurements, such as light, temperature, moisture? So basically taking all the environmental dynamics into account.
They do clearly affect it. Similar to drone measurements, we try to concentrate. That's why we also, among others, invest in such kind of positioning system to get quickly over under as steady state conditions as possible. If experiments are larger and you cannot always do that, we do include our light and temperature as variation over time, and we have our randomized designs. So it's two-sided answer. Yes, they influence our signal, and it should not be ignored. It should be quantified.
Mm. Maybe a question to your take-home message or your random thoughts. So you mentioned this, that there is not only one leaf, but there is obviously a canopy, and you should take this into account. But it's an important point to address. How would you address that?
Yeah.
How you,
No, good point, Colin. And it is. I mean, if I address it, as I said, in the one thing is that you could say almost sometimes blindly what we do is we measure with an excitation pulse of a few centimeters, 5 centimeters of the lift, that depending on the canopy that we're looking at penetrates deep or not into the canopy, and you will get an integrated signal of your canopy. We repeat these measurements many times over the canopy, and we call that canopy fluorescence. I think we're pretty close at an estimate what the canopy is producing, like it would get sunlight. Does that mean that we have a full understanding? No, there we're working on combining these measurements with 3D estimations, where the angularity and the penetration of light inside the canopy gives us further information. These are classical models are also built on leaf angles. Although if you scale up, you go back to a single leaf model, and we try to combine these 3D reconstructions with our photosynthesis measures at the moment. There's some excellent work there by Francisco Pinto, who combines sun-induced fluorescence with 3D reconstruction. And we are at the moment with the active fluorescence, we're looking in combination of the lift with another sensor, the so-called plant eye. But this is work in progress.
Mm-hmm. Okay, thanks a lot. So maybe moving already now back to questions to both of you. So there was one question, or let me first move back to the questions how the environmental factors affect the measurements. So that's probably also relevant for Shawn, for your measurements. Obviously, you want to understand plant environment interaction. You are measuring, having a different system, RGB. What's your impression, this environmental dynamics? How does it affect your measurements?
Well, we saw, I think I mentioned earlier, the sun angle and the amount of shadow having a big effect on it. I mean, otherwise, these indices, if you take them with indirect light, they work fairly well. With RGB, because it's broadband, sort of general reflections, it's not as sensitive as say a scientific multispectral sensor or something like that, where you really want to have direct sunlight and get better quality, spectral quality. You don't want to have wet leaves, I guess, but you know, these are sort of they're a snapshot, but it's not something that's going to change on a dynamic scale as quickly as photosynthesis or fluorescence, like Onno's work. And so, you know, you take the picture one day or another day, there's not going to be a big change in biomass from one day to another, but a couple weeks apart, you will see differences in canopy cover and greenness, but it's not so dynamic.
Maybe also another question to you, Shawn, to give Onno also some time to breathe after half an hour of talking. There was questions from the first session, which specifically from flight to data processing, which you actually responded in writing, but it might be relevant for everyone to briefly address that. So from flight to data processing, how long does each step take? So minutes, hours? So what's so it's. Yeah.
So we did some sort of, you know, back of the envelope sort of calculations, and, you know, drones become more efficient when you start talking about the scale of your phenotyping operation. And so if you have, we had one study that was like 140 plots. You get better data and better results if you just walk through the field and take a picture with your hand, because there's no pre-processing. You take that, you can set it up to process, and you'll be done. During your lunch break, you could do all the phenotyping. If you have thousands of plots, then you might want to do a drone. You could fly it, maybe a 20-minute flight, an hour or two to process, depending on how good your computer is, and then an hour or so to extract. So you could collect data just before lunch around solar noon, and then be done by the time you go home in the afternoon.
Mm-hmm. Okay. Thank you. So there is question to both speakers. So can remote sensing tools be used in field where the geology is not uniform? For example, one part of the field is lower than another. Does this create problems for data processing? So Onno, would you like to start?
Yeah, I saw Shawn also answer that, and that's the obvious thing where you have indeed interaction with a drone that is easier in terms of topography, but certainly also should be considered in terms of topography if you keep a fixed distance from about sea level or from the topography below you. In our, if I go more to the area that I've shown you on ground-based vehicles, we can cover a certain amount of slopes and changes in topography where we keep our sensors to a fixed distance to the plants. Nice things about plants is they always grow straight to the sky. So either you have them on the slope of 45 degrees, a plant will still be up straight. So in that sense, if you have your sensor in that oriented to your plant in the same angle, that's doable. It's more difficult indeed to drive up or push up and down a slope. In most phenotyping experiments, I must say also, and also the breeders large scale test sites, they actually choose rather flat sites because they want to compare and they know if you're more up to a slope or you have a south or north facing slope, you get a different result. So we don't typically face these problems so often. In recent agriculture developments, whereas for example patch agriculture, where we look more in detail on heterogeneities in field and try to apply, for example, to work with the heterogeneity of a field, this may become more relevant again and having vehicles driving through. But in general, also I could say again, if it's about agriculture, then I exclude natural ecosystems. The topography is often not limited because you can drive through with most vehicles in this field. Otherwise, they were not used for agriculture.
Shawn, do you want to add something to the topography?
Yeah, well, like I mean, if it's phenotyping, you know, think about how the geology is going to affect your phenotyping trials. Maybe more important than whether or not your technology is going to be adaptable. If it's on a drone, I sort of mentioned what I wrote down. Maybe you'll have a small change in resolution, but that doesn't really affect data processing. You know, if your spatial resolution of your pixel goes from 0.25 centimeters to 0.26 centimeters because your field's a little bit lower, that's not going to have a big difference. I don't think on the result.
Mm-hmm. There is also a question. What is the phenotype repeatability from day to day, from day one, day two? Do you have any results on that? So it would be first to Shawn and then Onno. So it's actually complementary work. So how repeatable is fluorescence measurements and RGB based measurements?
We're going to have good repeatability from one day on the end. I mean, unless the sky conditions or the cloud conditions, the environmental conditions change a lot. Like I said, the RGB is not capturing these minute dynamic differences that have to change over a fast scale. So if you do half the plot one day and half the plot the next day, there's not going to be a big difference. But it's fairly consistent results.
Mm-hmm. It's a little bit the origin from the question, but if we talk repeatability in terms of heritability, broad sense, then we do have indeed not we talk about this FQFM about 70%, 0.7 of broad sense heritability, right numbers on top of my head, that we went into. That's more the statistical part of repeatability. So yes, we do have data on that, and that's where I would like to refer to the work of Nicolas Sendonadi, who will indeed include these numbers of repeatability. And we got pretty high there actually with photosynthetic efficiency measurements over time.
Yep. Okay, thanks a lot. Looking at the time, so maybe the very final question to both of you. So plant phenotyping in 5 years, what is the game changer for you?
Yeah, for me, I said it already. Phenotyping is the data communication. We talk a lot about data storage and data reusability in the context, but I do think there's a large world out there. Did you see the people doing large and very interesting metadata analysis? But I do think that is one way, but that requires another kind of people almost to make use of all this data collected. For me, the other advantage of data storage and communication is that we can have a direct use of it and a direct communication where I asked Shawn, for example, already, would the height be possible as a parameter? Because I need to measure at a constant height for phenotyping. But you could also see a harvest that you would like to do at a specific height, or a flower you want to cut off or something. So that's what I see in 5 years' time in phenotyping, where data communication is really a center for all of us, and that allows us to think in more traits and data availability in real time.
Yeah, I think right now, with drones, we've suddenly become able to collect more data than ever beforehand, and I think we've got sort of backlogs of data that are building up now. But I hope that data processing to support decisions will be something that'll really catch up with the big data that phenotyping is collecting now, and will help us go from data to useful information a little bit quicker.
Okay, thanks a lot. So by that, I would like to thank the speakers. Shawn, Onno, thank you both for the presentation. It was really great. We had all the participants, they stayed to the end. Let me also close with a final slide. So basically something very general. So stay tuned. We will continue in the next year. So we are going right now into the winter break, and we'll continue in January, potentially February. There is also, since in between there are no webinars, you may have some time to address the survey. There is a survey on plant phenotyping. It takes 10 minutes roughly to get your idea, your impression on plant phenotyping. That's very important and valuable for us to see where we develop as a community, and to see what happens within the next year. What do you think about plant phenotyping and as such? So thank you once again. It was a very successful, not only the session today, but within the last half a year. So there was quite a number of people that joined the webinars, and we addressed many important topics. And as such, I would like to share a video with you and show you our facility, not only the facility, and wish you a Merry Christmas, going now into the holiday season. So and enjoy the time. And we are looking forward to see you in January or February. Let me play it once more. So this is our new facility, and it's not only for crops. So thanks a lot, and hope to see you soon.
Thank you, Ron.
Enjoy the holiday season, and see you.
Yeah,
You well.
bye. Thank you, Onno.
Bye-bye.
Thank you, Shawn.
Thank you. Bye.
Bye. Warte mal, Roland.
Ja.
Ich mach mal eben noch unsere Rollen wieder zurück.
Rollen?
Machst du mich mal eben wieder zum Host?
Warte mal, wo bist du denn? Wir haben noch ein paar Leute drin.
Ja, ist egal. Das ist wurscht. Ja, genau. Und jetzt nochmal zum Host benennen, bitte.
Also Host erstellen, ja.
Ja, genau, super, Dankeschön. Bye bye everyone, have a nice weekend.
Bye bye.
Bye bye.